All right, so pull this up. So we left off talking about enzymes and the very important role that enzymes play for a number of reactions in a cell. Okay, let's talk about some more characteristics of enzymes. So enzymes are proteins, right? Or most enzymes are. And when we talked about proteins that they have um, multiple levels of structure. Okay, and so enzymes themselves are three-dimensional. They have that hierarchy of structure: primary, secondary, tertiary, quaternary levels of structure that proteins have. So they're three-dimensional. Okay, and so the reactant or reactants that enzymes work on are called the substrates. Okay, all right, and so because the enzymes are three-dimensional, the enzymes have a site that the substrate or substrates bind to. Okay, and that site is called the active site. Right? And so only substrates that fit the active site will properly bind. And that's this idea of the lock and key theory. Okay, so you have the key and you have the lock, the enzyme. Okay, the substrate's the key. Only the right key substrate will fit into a particular enzyme. All right, and so most enzymes are very specific for the substrates that they work with. All right, and then it's also this idea of induced fit, so that they not only, when the substrate binds to the enzyme, all right, um, when they bind, when the substrates bind to the enzyme, it induces the enzyme to change its shape ever so slightly to create an even tighter binding between the substrate and the enzyme, and that's the idea of induced fit. All right, so when the substrate binds to the enzyme, all right, that process of binding or the action of binding will cause the enzyme to change its shape to produce an even tighter binding between the substrate and enzyme, and that's the induced fit. But when the substrate binds to the enzyme, then the whatever chemical reaction happens will occur. All right. Um, then the substrate or substrates are then converted in that chemical reaction, and then they are released. And then the enzyme again is not consumed by the reaction. All right, it releases the products, and then it frees itself up to be able to bind another substrate and participate in another chemical reaction. All right, so when an enzyme and a substrate bond together, all right, for that whatever chemical reaction to occur, the enzyme will then release the products from the chemical reaction. All right, and then that active site is open again for another substrate to come in and bind to the enzyme. All right. Well, there are a number of things that can affect enzymatic activity and how quickly the enzymes can work. All right. First thing are enzyme and substrate concentrations. All right. The more enzymes, the faster rate of enzymatic activity will occur. Right. Okay. So if you have more things out there that are able to interact with substrates and convert them to products, the faster these reactions will occur. All right. Same thing on the substrate side. Okay. The more substrates you have in a certain area, okay, all right, that are able to bind to the enzymes, the faster the reactions will occur to get the products. All right. Now, but that is not an infinite relationship. you can, let's say you have the substrates at a specific level, at a constant level. You can't just continually increase the enzymes more and more and more to get faster enzymatic activity. Why not? Well, eventually you're going to react with all the substrates that are there. All right. And so there's nothing left for that enzyme to react with, or those enzymes to react with. So in that instance, the substrate is considered the limiting factor. Okay. All right. So because the enzymes can only work if there are substrates available. So if they go ahead and convert all the substrates that are available to react, then enzymatic activity is going to go down to zero. So in that instance, substrate is the limiting factor. Vice versa, if you increase substrate concentrations, all right, to increase enzymatic activity, all right, then that's fine. But eventually, all right, all those enzymes that are kept at a constant level, they're going to be busy. They're going to be full of substrates, all right, and so there won't be more enzymes to convert the substrates in the products. And in that instance, the enzyme is the limiting factor. Okay. Temperature can affect enzymatic activity. Increasing the temperature will increase the enzymatic activity. All right, because when you increase temperature, the molecules move around more quickly, and that's going to increase the instance of the enzymes and the substrates coming into contact with one another. All right. However, again, that's up until a point. All right, because what are enzymes again? They're proteins. All right, and so what happens when you increase the temperature to a point where that protein can no longer take it? You remember, it loses its structure. All right, and what's that process called? Denaturing. Okay, and so if the temperature gets too hot, that enzyme is going to lose its structure, become denatured, and when it becomes denatured, it can't function. All right, so high temperatures can denature an enzyme, and when they denature an enzyme, it will stop functioning. Okay. So there's an optimum range for temperature of enzymes. All right. Now, same thing with pH. There's an optimum pH range. All right. What do you think the optimum pH range is for enzymes in our body? Well, it's going to be around neutral, around 7, right? Because our body stays around pH 7. All right. And so if you get out of that optimum pH range for enzymes, then they will again become denatured. If you get too basic or too acidic, and an enzyme can't take that, it will become denatured and stop working. So, but enzymes can have different optimum ranges. All right, enzymes that are in our body, okay, have an optimum range around 98.6 Fahrenheit. All right, but enzymes that occur in organisms that live in deep vents in the oceans that are much hotter environments, all right, they can take much hotter temperatures. So their optimal range for temperature is higher than what it is for us. Okay. All right. So this kind of graphs it out. All right. And so here is optimum temperature for our enzymes, okay, where the optimum temperature is around 98.6 Fahrenheit or around 37 degrees Celsius. All right. If you get too low, then they're going to kind of slow down and not really work well. If you get above this optimal temperature, okay, they start to break down. All right. Now, with thermophilic bacteria enzymes, okay, all right, bacteria that love hot environments, their enzymes have a different optimal temperature range, which is much higher. Okay. Now let's look at enzymes that can be found in our stomach versus our intestines. Our stomach is a very acidic environment, and so the enzymes that are working in our stomach is pepsin is the main one. This optimum pH range is pH 2, very acidic. All right, now you have another enzyme, trypsin, which works in our intestines. All right, its optimum pH range is much more neutral to a little basic. All right, it would break down in the high pH of our stomach. All right, so two enzymes, all right, working in two different environments have different optimum ranges for pH. All right, some enzymes require accessory molecules to function properly. Okay, and so, and without those molecules, they may not be able to work at all. All right, so there are two types of accessory molecules. You have cofactors and you have coenzymes. So if those helper molecules are non-organic, they're called cofactors. All right, and examples include several metal ions, magnesium, manganese, nickel. If those accessory molecules are organic, they are called coenzymes. All right, and vitamins are an example of coenzymes, such as vitamin C. But as you can see, they don't bind to the enzyme at the active site. All right, instead, they'll bind to the enzyme at some other spot. Okay, and help the enzyme work properly. Okay, all right. Now here, if they do sort of bind to the enzyme in the active area, they won't bind at the same spot as the substrate. So ATP, there we go again, adenosine triphosphate again, is a special coenzyme. It functions as a coenzyme in many reactions. Okay, it bonds between the bonds between the phosphate groups in ATP hold a lot of energy, and so when a phosphate group is transferred via a process of phosphorylation, energy is transferred along with it. All right, and so ATP will transfer a phosphate group to an enzyme and give that enzyme energy to go have through a reaction. All right, and we said so the ATP to ADP. So the process by which cells generate ATP, okay, is this ATP ADP cycle. All right, and adenosine diphosphate forms when a phosphate group is removed from ATP. Then ATP excuse me. Then ATP forms again as ADP gains a phosphate group. Okay, so again, it's a cycle. Okay, when ADP gains a phosphate group, it becomes ATP, and when ATP loses a phosphate group, it becomes ADP. All right, and this cycle couples endergonic reactions with exergonic ones. All right, so here again, it's just saying that that this ATP, okay, there's a lot of energy that's in these bonds, okay, in these chemical bonds, and a lot of times when this ATP loses this phosphate group right here, this bond is broken, then energy is released. All right, and what we see is, and what we've said before, is that this cycle is coupled, okay, with other endergonic reactions, so that to fuel those endogynic reactions. All right, so ATP is an example of a coenzyme. All right, so some molecules try to slow down enzymatic activity. All right, and so we get this idea of allosteric regulation. Okay, so allosteric regulation, it could be an example of activation. Okay, and so when you have allosteric activation, the active site becomes available to the substrates when a regulatory molecule binds to a different site on the enzyme. All right, so the idea of cofactors and coenzymes, that gets to allosteric activation. All right, but then you can also have allosteric deactivation. So when a regulatory molecule comes in and the active site becomes unavailable to the substrate, when that regulatory molecule binds, then that's an idea of allosteric deactivation. All right, and so competitive inhibitors is an example of allosteric deactivation. All right, so competitive inhibitors compete with the substrate for access to the enzyme's active site. All right, so you see here, all right, the substrate will bind to the active site. You have the chemical reaction, all right, and then you have the products. Well, with inhibition, all right, you're going to slow down enzymatic activity. All right, and so a competitive inhibitor will compete with the substrate to bind to the active site. And as long as that competitive inhibitor is bound to the enzyme's active site, the substrate can't bind to it. All right, and so when the active site is taken up, then it's going to slow down enzymatic activity. So the competitive inhibitor competes with the substrate for the enzyme's active site, and this will slow down enzymatic activity. All right. Then you have non-competitive inhibitors. They don't compete with the substrates for the active site. Instead, they bind to the enzyme at a spot somewhere different than the active site. All right, and that binding creates a conformational change in the active site, thus preventing the substrate from being able to bind. All right, so you see here, all right, the active site, substrate binding to it. Well, a competitive inhibitor is going to compete with the substrate for the active site. A non-competitive inhibitor is not going to compete with it. Instead, when they bind, they cause a change in shape in the active site such that the substrate can no longer bind. All right, so the competitive and non-competitive inhibitors both decrease enzymatic activity, but they do so in different ways. All right. The end product of a series of enzymatic reactions often inhibit the activities of one of the enzymes in the series. All right, so you get this idea of feedback inhibition. So here you have a series of reactions that go from the original reactant to the product. Okay, and what we see a lot of times in nature is that at some point when you have enough products, they will go and inhibit the enzymatic activity of one of the enzymes in the pathway. And this is the idea of feedback inhibition, which is a regulatory mechanism. It's a change that occurs during a specific cellular activity that suppresses that activity. Okay, in this instance, products will come and feed back onto this pathway and inhibit this particular enzyme so it prevents more product from being produced. Okay, sometimes too much of a good thing is a bad thing. All right, and so what happens is that it's evolved this path or this process by which products will then feed back on an enzyme in the pathway, inhibit to slow the overall reaction down. Okay. So here you go. Okay. All right. So you are again you are slowing this pathway down. Okay, such that you're going to get fewer and fewer products being made. All right, so in feedback inhibition, the end products of the pathways is able to turn off some step in the pathway, and it's important because it prevents an overabundance of end products. All right, which could be harmful if it continued unchecked. So feedback inhibition slows down enzymatic activity, and it prevents an overabundance of end products. Okay. All right. So, need to know what are different types of energy we talked about, potential kinetic weather difference. What's the difference between endergonic and exergonic reactions? All right. How do cells deal with these reactions? How can you identify a chemical reaction as being endergonic or exergonic? All right. How does the cell use energy coupling? Okay. And what is that concept of energy coupling? What are enzymes? What are the importance of enzymes? All right. How do they work? All right. And what are the different factors that we talked about that can affect enzymatic activity? All right. And how do they do that? All right. Which ones speed up enzymatic activity? Which ones slow them down? And how do they do those things? Okay. All right. So if you have any questions, just let me know, and just keep going, keep at it. All right.